All right. Good evening. Today I'll not be showing my video due to some issues that I'm having at the background at the moment, but I hope it's comfortable with you. So I hope that you have done the paper as well. Have you done
Yes,
it with the correct timing?
Yeah, yes, miss.
All right. Then let's go for the discussion.
Yeah.
Okay. Right. So this is the 2023 January biology paper that we are discussing today. So in this paper, you can see that they have given a description, right? This is the second paper that you are getting. So they have given a big description. And the most important thing over here is you don't have to read everything in this description and waste your time, right? That means first I think it's better if you go with the question. First read the question. I think this is a method that is being taught in English as well. Whenever you get an essay, right, first thing you need to do is you need to read the questions that they have given under that essay, so that you can have an idea of what they are asking at the moment. Then you can go back into this essay that they have given, and then you can select the important points, right? That is to save your time. But if you are a fast writer, right, if you usually write your paper fast, then it's okay to read this and have an understanding and then go for the questions. Doesn't matter, whatever you do, it should be suited for you, right? So what I did was, during my times, I used to do the questions, and I used to come to this particular paragraph and take the points out from there. Plus also, the questions are not always based on the essay that they have given, right? They're asking the questions from the things which are in your course book, right? In your biology course book, the syllabus. So you have to have an idea about those things, not basically about this particular essay. Right. Anyways, I'm not going to read this essay because that would take a lot of time, right? So I'll go with the questions, and that would be easy, right? So you can see in the first question, they're asking, explain why plants have been dug up and transported at most risk of drying out. You know, when plants are uprooted, right? When plants are uprooted and when they are being transported, it damages the root hair cells, right? Because these are the roots, and in these roots, you can see tiny root hair cells like this. You can't see, you have to see using a microscope. You can see tiny root hair cells like this, right? So these root hair cells, they increase the surface area for water and nutrient absorption, increases the surface area for nutrient and water absorption. So now when these trees are being uprooted like that, right, especially when they're being dug up, right, using a shovel or something, you dug it up. So what happens is these root hairs get damaged. That means their surface area is going to get reduced, right? Surface area will be reduced. When the surface area is reduced, can they take in the amount of water that they used to take in before? No.
No.
Right? Yeah, no, because the surface area is reduced now. But you know, if you take a tree, there is a process of transpiration, process called transpiration. Transpiration means the loss of water from the plant leaves as water vapor, right? Water is lost into the surrounding air as water vapor using diffusion, right? Diffusion. And mainly they use the stomata for this, right? They have cells, they have a special type of cell called guard cell, right? A special type of cell called guard cell, which makes up this stomata. Stomata is just a space in the plant. If you take the cross section of a plant leaf, you know the epidermis is like this. Then you know that the palisade layer is like this. Then you know the spongy mesophyll, it's like this. And then you know the lower epidermis is like this. Did you know that the lower epidermis has a lot of stomata in it compared to the upper epidermis? There is a reason for that. Now we are
Yeah.
not going to learn what that reason is, but anyways, I just wanted to give you an idea of what the stomata is. Transpiration happens through the stomata, right?
Yeah.
So when you dug up something, when you dug up the plant, the root hair cells are damaged. That should be your first point, right? Should be your first point. Then you have to mention that water is not absorbed or taken up. You can write it in your own words. You can say that enough water is not taken up or enough water is not absorbed. You don't have to write the exact marking scheme point, because in the marking scheme they say an answer which includes these 3 points, right? So it should be a description. So you should have these 3 points in your description. So you should mention that root hair cells are damaged, and because of that reason, not enough water is taken up when compared to the water lost by transpiration. Now, if we lose a lot of water, when compared to the amount of water we drink, we'll get dehydrated, right?
Yeah.
Will get dehydrated. Just like that, the tree can also be dehydrated, right? That is why they say it dries out or it wilts, right? It wilts. The plant will wilt. Therefore, this is the complete answer. If you have any 2 of these 3 points, you get 2 marks, right? Root hair cells are damaged and water is not absorbed or taken up. When compared to the water which is lost by transpiration. Okay?
Yeah.
Right. Now let's move on to the next question. You can say it's nothing to deal with the above. The first question was nothing to deal with this first paragraph that they have, the essay that they have given. If you go to the B, you can see they're asking us to determine the number of stomata per millimeter square on this leaf surface. Now, as soon as you see this diagram, you should understand how many stomata are initially there in this given diagram. Can you see these bean-shaped cells? The bean-shaped cells, yeah, they are the guard cells, right? They are the guard cells. Covering, that means these guard cells are enclosing the stomata. In the middle of these guard cells, you can see small holes. They are called the stomata. Stomata is the area where gaseous exchange take place, right?
Yeah.
So, yes. So in this diagram, you can see 3 stomata. That means 3 stomata in this diagram means you should find the area of this diagram. They have already given us the dimensions, right? Now we just have to calculate the area. Since they're asking us how many stomata are there per millimeter square, it's better for us to convert these micrometers into millimeters so that we won't face any errors when doing the conversions after getting that big value, right? Final value, rather than converting our final value into the required units, it's better if we can convert them in the first place itself. Because at the end, you'll have to deal with micrometer squared into millimeter squared. Sometimes due to exam stress, this could be a little bit not hard, but it could be a little bit challenging and it could stress you out. Because of that reason, I know it's easy, but because of that reason, it's better to do the conversion first, right?
Yeah.
So, yes. So you can see 100 micrometers converted into millimeters. Let's do it. 100 micrometers converted into millimeters means 1 micrometer, 1 millimeter is equal to 1000 micrometers. 1000 micrometers, yes. So 100 micrometers means 0.1 millimeters. So therefore, the dimensions are 0.1 millimeters over here and 0.1 millimeters over here. So how do you find the area? You multiply the length by the height. So 0.1 millimeters multiplied by 0.1 millimeters, you get 0.01 millimeters squared, right? 0.01 millimeter squared. So they're asking us to determine the number of stomata per millimeter squared. How many stomata are there? There are 3 stomatas, right? Yeah, 3. The plural of stomata is stoma. Okay, plural of stomata is stoma. There are 3 stoma. So you can see 3. That means 3 are there in 0.01 millimeter squared. Then how many are there in 1 millimeter squared? That's what we are needed to find over here. So to convert 0.01 into 1, we have to divide 0.01 by 0.01. That's how we get 1 millimeter squared. So if we do something to the right hand side, we need to do the same thing to the left hand side as well. So I'll be dividing this by 0.01. So 3 divided by 0.01 will be 300. So how many stomata are there? 300. Yes, number of stomata per millimeter squared will be 300. So here, if you have taken the final answer even without showing any calculations, here they have given you marks in this particular marking scheme, right? But what I advise is it's always better to show the steps clearly, because sometimes the examiners could be different. That means this year's panel will not be the next year's panel, right? So it's always better if you can show the particular what do you call the way that you have done it, right? In that means the steps of getting the answer before writing the final answer, right? All right, let's move on to the C1. Here they have asked, explain why stomata regulators reduce photosynthesis. And look how easy this is. They have given us the lines to read as well, from line number 19 to line number 24. Right, let's just have a look at that. Line number 19 to line number 24, this part. See, I have already highlighted that part. Let's see. Okay, the second type of antitransparents are stomata regulators. Antitransparents means something which you use to reduce the transpiration. That's what you mean by antitransparents, right? So if you want to reduce the water loss from a plant, you can apply any antitransparents. So they have already talked about a type of antitransparent over here, which was not required for any of the questions that was asked in the paper. But now the second type of antitransparents are important for us. What are those? They are stomata regulators. That means they control the size of this opening, right? You can see they reduce the size of the stomatal pore. Now, you know this guard cells can control the size of the stomata. If the guard cells are big and turgid, right? If they are big and turgid, you can see the stomata will be open like this. But if the guard cell is very small, you can see the stomata is closed like this, right? If the guard cell should be shrunk, actually like this, shrunk. Then the stomata is small, right? So basically, so basically the second type of antitransparents, they reduce the size of the stomatal pore. And you know, when this stomatal pore reduces, the water lost by transpiration will be lost, right? Will be reduced, right? You know, in a plant, these stomata are the structures where the gaseous exchange takes place. That means water vapor is lost, carbon dioxide is taken in for photosynthesis, oxygen is given out as a byproduct of photosynthesis. For respiration, again, oxygen is taken in. As a byproduct of respiration, carbon dioxide is given out, right? Because both photosynthesis and respiration takes place in plants, right? So all these gaseous exchange take place via this stomata. right? Due to that reason, if you reduce the size of the stomata, it won't just affect the movement of water vapor, but it would also affect the movement of other gases like carbon dioxide and oxygen. So here they have said, here you can see stomatal pores affect diffusion as well as transpiration. Diffusion means basically transpiration also happens due to diffusion. Movement of
Yeah.
water molecules from a region of high concentration to a region of low concentration. In Edexcel IGCSE, they say from a dilute to a concentrated area, right? That's
Yeah.
how they say it in Edexcel IGCSE. So you can see photosynthesis and growth may change when stomatal regulators are applied to the leaf surface. Why? Because when the stomatal size reduces, only less carbon dioxide is taken in. We know that the plants require carbon dioxide for photosynthesis. So if they take in less carbon dioxide, the rate of photosynthesis will decrease, right? Photosynthesis is the process where they make the required food for the plant, right? Where they make the required food, the chemical energy, right? Therefore, if photosynthesis reduces, the plant growth will also be eventually affected. Due to that reason, these antitransparents have a negative effect on the growth of the plant, but it also has a good effect. Yeah, it also has a good effect. Why? Because it will prevent the excess loss of water from the plant. So we need to make sure we always apply only a certain, we not apply, we administer only only the required amount of antitransparents to that particular plant, right?
Yes.
Now you can see over here, they have said, explain why stomatal regulators reduce photosynthesis. Even though they have given a lot of lines, right? The expected answer, it's better if you can explain it, right? It's better if you can explain it. The expected points are stomata is closed, or else you can write the stomata pore is reduced. Stomata pore is reduced or made smaller. Stomata pore is reduced or made smaller. You can write something like, yes, you can write something like this, and you can write, therefore, less carbon dioxide is absorbed. Now, since it's better to write better, not actually, what matters is the quality of the answer over the quantity. So it doesn't matter even if your answer is short, right? But it's better if you can write that taking in less carbon dioxide would affect the rate of photosynthesis. So basically you're supporting the question that they have asked, right? That means at the end you're giving the question a bit of a support, because when you say less carbon dioxide is absorbed, like the examiner wouldn't know whether you know that carbon dioxide is needed for photosynthesis or not, right? Maybe you know, but then that's why you wrote it. But it's better to always write a complete answer saying that taking in less carbon dioxide would reduce the rate of photosynthesis, right? Okay, let me know if I'm too fast for you. Today I'm not speaking a bit loud because I'm not at my house, right? So let me know if I'm too... Okay, right. Let's go to the D1. Here they have asked, explain why reflective compounds should only be applied to the upper surface of a leaf. Now, it's not a hard question. They just have given us a new word called reflective compounds. But actually, the answer is not in that essay that they have given. It's just the word reflective compounds is new for us. So we can go over there and we can read what these reflective compounds are. But the answer is not in that essay. Answer is in the things that you have learned in your course, learned from your coursework. So you can say this is the answer. I'll be explaining the answer, but let's read line 27 and 28. Since this is the first day of doing the class, it's better to read the lines and then go for the question, right? Answer. 27 and 28. You can see yes, you can see the coating is only applied to the upper surface of the leaves, so does not block the stomatal pores. Coating a leaf with a reflective chemical can reduce leaf temperature, transpiration rate, and also increase the efficiency of water use. Right. Now, sometimes when you're doing the paper, if you didn't read the essay, you might be a bit insecure about your answer. So if you think you need to read the particular essay that they have given, it's always better to do so, but always give priority to the questions as well. You can see here they have said they're applying some reflective compounds on the leaf surface, right? When you apply these reflective compounds, the gaseous exchange is blocked, right? Because the stomata is covered, right? Now imagine on the top of the leaf, if there are any stomata, right? Stomata are not like this. I just wanted to show you, right? You can see on the top of the leaf, there are stomata. So you are applying a coating over that, right? Which are reflective compounds, right? So what happens is gaseous exchange will be blocked, but they're asking why they are only applying it to the upper surface of the leaf and not to the lower surface. The reason is because the lower surface contains lot of stomata. That is the point that you should know. When compared now, when actually if you take the cross section of leaf, the cross section of the leaf is like this. There is a epidermal cell layer like this, right? Epidermal cell layer. In between there can be one or two stomata, right? I'll draw a stomata like this, right? Then there is a layer called the palisade layer, or you call it the palisade mesophyll. They are long elongated cells like rectangles, and there you can find chloroplast. And remember, these guard cells are the only cells in the epidermis which contains chloroplast. Other cells in the epidermis, they don't contain any chloroplast, but these cells, they do contain chloroplast. These are the palisade mesophyll cells. Palisade mesophyll cells. Then next you have a layer of cells called the spongy mesophyll. You can see there are a lot of air spaces between the spongy mesophyll layer. And below to that layer is finally our lower epidermis, which means the lower surface of the leaf. So basically it's like this. There are a lot of stomata in this lower epidermis of the leaf. See, there is one stomata over here, another stomata over here, another stomata over here, another stomata over here. So basically when compared to the upper epidermis, if you take a look at the lower epidermis, the number of stomata in the lower epidermis is considerably higher. Because of that reason, if you apply reflective compounds on the lower surface, important gaseous exchange will be lost, right? For example, carbon dioxide needs to be absorbed, right? Because why? The plant needs to carry out the photosynthesis. We need to somehow block the transpiration, as well as we need to carry out at least a bit of photosynthesis, right? Because of that only we are not applying the reflective compounds onto the lower surface. By applying the reflective compounds onto the upper surface, we can at least reduce the rate of transpiration by means of, that means not by a large amount, but by little amounts, you can reduce the rate of transpiration, right? It won't help us a lot, but yes, that would be beneficial because at least you apply it on the upper surface. Why don't you apply it on the lower surface? Because there are a lot of stomata, and means mainly gaseous exchange takes place via those stoma. So because of that reason, we are not applying any reflective compounds to make sure that the plant will still carry out the photosynthesis. Therefore, this is the answer that you need to write. Even though it says 2 points, they have given 4 points in your marking scheme. And out from these 4 points, if you have any 2 points, you can get your marks, right? But you need to make sure
Yeah.
you write you need to make sure that you write a complete answer to this question, right? So you can see I have written stomata is mainly or only present on the lower surface. It's like this. In some plants, they don't have any stomata on the upper surface. Mainly stomata are present only on the lower surface. But if you compare certain plants, they do have stomata in both the upper surface as well as the lower surface. Right? There is a type of plant called xerophyte, not in your syllabus, right? You might have just heard about it, right?
Also, water lilies as well. They have stomata on the outer surface, I think.
Sorry, I didn't hear you.
Yes, water lilies. I think they also have stomata on like on the outer surface.
Water?
Water lilies.
Watermelon leaves.
And it was water lilies, or was it water lotus? I have no clue.
I am not really sure. Can you please text me that?
Yeah,
Later.
sure.
Later, okay? Right. Okay. So, you know that there are these plants called xerophytes. These xerophytes, they are the ones which are living in the deserts, right? These desert plants, actually, they also have stomata mainly on the lower surface. They don't have any stomata on the upper surface. And if you take a look at their stomata is sunken like this. Their stomata is sunken. The reason is it helps to reduce the rate of transpiration then, right? But that's just a bit of extra knowledge. Let's get back into the question. Here they have said the answer should contain stomata is mainly or only present on the lower surface, right? Next point should be stomata is not blocked or covered by the reflective compounds. So basically, when you apply the layer of coating onto the upper surface, the lower surface stomata is not blocked by the reflective compounds. Therefore, still carbon dioxide will be absorbed, right? And you can see more light falls onto the upper surface, right? That means these reflective compounds are there to make sure that the light does not damage the cells. Sometimes light, the harmful rays in the light, for example, UVA, UVB, these could actually damage the interior of the plant. To make sure that they won't damage only, we are basically applying reflective compounds. We also apply it as a means of anti-transparent as well, right? So if you have any two of these points, you get 2 marks, right? It's better if you can explain it, right? Let's move on to the next. Please send me the name of the plant that you said, right, later, so that I can have a look at that.
Yes, please.
Explain why reducing leaf temperature reduces the transpiration rate. So basically, even though they have said it's from lines 28 to 32, you can think of this. Now, you know, when temperature is reduced, it reduces the kinetic energy. The energy of the molecules decreases. When the temperature is increased only, the molecules start to move very fast, right? But if the temperature is decreased, the molecules start moving very slowly. As a result, you know, they move out through this stomata very slowly, right? Therefore, what happens to the rate of transpiration or rate of any process which is taking place? Rate of any process will be slow, right? It will be a very slow rate. What is the reason? Because the molecules don't have enough kinetic energy. Due to that reason, they can't move very fast out through the stomata. In that case, it will reduce the transpiration rate as well. Not only transpiration rate, it can also reduce the rate of photosynthesis. Why? Because carbon dioxide molecules also, they also have a very less kinetic energy, right? Because of that reason, now when the carbon dioxide molecules come inside the leaf, they will have a very small temperature. That means they have very less kinetic energy. As a result, They want that as a result, the photosynthesis rate will also be slow. But here they're talking about the transpiration rate. So you must mention lower temperature. What happens when there is a lower temperature? You can't directly write lower temperature. Because of lower temperature, water molecules move less fast, right? You can't write like that. You must mention the energy because they expect the point energy from you. So you must say the lower temperature will reduce the kinetic energy, right? And then you must say, therefore, the water molecules will move less fast. And therefore, less rate of diffusion means less rate of transpiration, right? Less rate of the diffusion rate is slow means that the transpiration rate will also be slow. Diffusion is the movement of molecules, basically, right? So movement of molecules from a dilute area to concentrated area. So basically in transpiration also that's what happens. Inside the leaf there are a lot of water molecules, outside not much, therefore they move out. So therefore diffusion, the rate of diffusion is very low at this moment. Why? Because the temperature is low. So if you have any of these 2 points, right, any of the aforementioned 3 points, you can see it's better if you can write this point. This point is really good. You must mention the energy. Plus if you have any of these 2 points, you can get the 2 marks, right? Okay, now let's move on to the next one. Let me know if you need any time to write down the answers, right? Okay, E1 you can see, describe the role of one named mineral iron in plant growth. In your syllabus, you are learning about 2 mineral ions which helps in plant growth, right? One is the magnesium and the other one is the nitrate. Okay, so you need to know magnesium is used to make the photosynthetic pigment called chlorophyll, which is inside the chloroplast, right? Which is inside the chloroplast of a cell. So if you don't have magnesium, if a plant doesn't have magnesium, that would cause the yellowing between the veins of the leaves, which means chlorosis. Chlorosis means the chlorophyll is not there properly. Lack of chlorophyll. That is what you call as chlorosis. And you can say nitrate. You know, nitrate ions are used to make amino acids, right? Amino acids. And amino acids can be converted into proteins. Why? Because amino acids, they polymerize like this and then only they create the proteins. So if you don't have enough, if the plant doesn't have enough proteins, even if we didn't have enough proteins, we would have a stunted growth, right? So plants will also have stunted growth. Not just that, they also will have the yellowing of the veins, right? So basically yellowing of leaves, right? So this is magnesium has yellowing of veins between the leaves. Veins, the veins are yellowed, right? That means between the leaf, that means in the leaf between the veins there's a yellowish color. But if you take the nitrate deficiency, the leaves are completely green, right? Completely yellow, right? That means almost many parts are not completely yellow. Most parts of the leaf is completely yellow, right? Not just specifically the areas in between these veins. So that is the difference between magnesium deficiency and nitrate deficiency. Here they have asked, here they have asked to describe the role of one named mineral ion in plant growth. Either you could choose nitrate ions or you can go with magnesium ions. So nitrate ions, they synthesize amino acids or proteins. You can mention that, right? If you want, you can extend it further, say amino acids or proteins which are required for growth. Right? In that case, you're writing a complete answer. But you get the 2 points if you have written nitrate ions to synthesize amino acid. And then you can write magnesium ions for chlorophyll. Because when you're writing the paper, you don't know what is the answer in the marking scheme. Right? So when you write the answer, it's better to write a full answer. Nitrate ions synthesize amino acid to prevent stunted growth, because they're asking the role, right? Role. That means even though they're not asking the deficiency, they're not directly mentioning us to, that means they have given a lot of space for us. And if we just say nitrate ion synthesize amino acid, it's not the way that we are supposed to answer that question. It's better if we can give more details, give more accurate details that we already know. So we know the deficiency, so it's better to write nitrate ions to synthesize amino acid to prevent stunted growth, or magnesium ions to make chlorophyll to prevent the deficiency between the to prevent the yellowing between the veins of the leaves. Then it's a complete answer, right? So if you have written any of the two ions and a function of that, you get the 2 marks, right? Then you can see in the F question, they're asking, describe how water is transported from the soil to the leaves in the plants. It's quite easy, right? You can see the root hair cells are, the roots are like this. And if you take one single root, they have a lot of root hairs, right? They have a lot of root hairs. A root hair cell looks like this. A root hair cell looks like this, right? It looks like this. So I have drawn many root hairs. These are microscopic. You can't see them, right? So you need to mention about the root hair cells, because they are the ones which increases the surface area for the water absorption, right? So it's better, otherwise they could have been just typical cells like this, right? Why do they have a special structure? The reason why they have a special structure like this is to increase the surface area, because when you have cells, when you have structures like this, it increases the surface area when compared to structures like this. So due to that reason, it's better to mention root hair cells increases the surface area for water absorption. Then you can write down how water is absorbed. Yes. Then you can mention how water is absorbed. You know that water is absorbed by the process called osmosis. Osmosis is a special type of diffusion. Actually, it is the diffusion of water molecules, right? The diffusion of water molecules. So you need to mention it absorbs water by osmosis. There is nothing different between, that means there is not much of a big difference between osmosis and diffusion. Diffusion is just the movement of any type of molecules. Osmosis is specifically the movement of water molecules. Yeah, the water molecules from a region of high concentration to a region of low concentration. But here, if you use the word high concentration, you can get confused. That is why they are saying dilute. Dilute means a lot of water is present. We say a solution is diluted. For example, if we take hydrochloric acid, we can say that is dilute hydrochloric acid. Why do we say like that? Because it has a lot of water in it. Or else, sometimes there are bottles in our laboratories, they are called as concentrated hydrochloric acid. The reason is they don't have much water inside that. So dilute means a lot of water is present. Concentrated means less water is present. So osmosis means the movement of water molecules from a dilute solution to a concentrated solution. It's better to see, they have given 1 point for explaining what osmosis is, right? They have given 1 point. If you miss that point, you need to explain these things to somehow cover up for the 4 points. So it's always better to explain each. That means wherever you are ready to explain, wherever you have the knowledge, it's better to explain. If you can say root hair cells increases the surface area for a water absorption, that is just one complete answer. It won't take much space either, right? Sometimes even though they have not mentioned the surface area, in another paper they could mention it. So it's always better to write complete answers, because you're writing the paper once, so it's always better to write complete answers. Then you can see water moves up by the xylem. After the water enters the root hair cells, right, there are so many cells that they have to pass. There are so many cells. Finally, they are entering the xylem lumen, right? The lumen of the xylem. Lumen of, yeah. Okay, lumen of the xylem. It's a hollow vessel, right? You know, it's a hollow vessel. Why? Because it's dead. It doesn't have any cytoplasm inside it, and it's lignified as well. Because of that, it's just a dead hollow tube, right? Xylem is just a dead hollow tube. So after water enters through the root hair cells into the xylem, through the xylem, water moves up like this, right? Why do the water moves up? Because can you remember we learned about transpiration? Water is lost as water vapor. Due to this creates a pull, right? This pulls this water column inside the xylem. This pull, right? Because now when water is lost like that, you need more water to fill the remaining gaps, right? Because of that, there is a pull on this water column inside the xylem vessel. We call that pull as the transpiration pull, right? So water moves up the xylem by a process called, or by a due to a force called as the transpiration pull, right? Transpiration.
Miss, also can
Yes.
we say it's transpiration stream?
Yeah, transpiration stream also, no issues, right? Transpiration pull or transpiration stream. And also this happens due to loss of water. You need to mention why this happens as well. Transpiration pull happens due to what? Due to the loss of water from the stomata, right? So if you have written any of the points which are given over here, you get 4 marks for that, right? You get 4 marks for that, right? Let's move on to the next question. I think it's better. Now I was thinking transpiration. If you have mentioned transpiration stream, you're meaning the movement of water, right, upwards. So there is a it's correct. It's correct. There is no issue in saying like that. But it's better if you can explain how that happens, like due to the loss of water from stomata, because of that only there is transpiration, due to that only there is a pull like this, right? It's better to explain. I know that you might have not written the word pull, but learn it now. It's better to write the word pull, but transpiration stream, if you have written, we can't say that it's wrong, because they're asking how it is being transported into the leaves, right? That means into the leaves means you have learned it as by the transpiration stream. So it's correct. But I think the most appropriate answer would be pull, right? Most appropriate answer would be pull. But if you have explained about transpiration, that also you can get 1 point, right? Because they have not, I need to double check the marking scheme. I'll confirm it after the class, right? I don't have the marking scheme at the moment. Because you have a doubt on that, I'll confirm it after the class, right? Okay. Then you can see the second question. Here they have showed us a kidney, the section of a human kidney, and they have labeled some parts in this kidney. So basically, you can see this outer structure is called as the renal cortex, and this inner structure is called as the renal medulla, right? You can see, yes, you can see there are tiny tubules like this, right? There are tiny tubules. Actually, over here only, inside these tubules only, our blood is filtered, or ultrafiltration process takes place in these tubules, and these tubules are called as nephrons, right? They are called as nephrons. That is not the answer. That is the answer. The what do you call these tubules are called nephrons, right? The nephrons actually look like this. They have a what do you call Bowman's capsule like this, and they have a DCT, distal convoluted tubule, and they have a loop of Henle like this, and they have You don't have to learn the DCT and PCT. You just have to learn there is a loop of Henle like this. So this is the nephron, right? This is the nephron. So you can see in the Bowman's capsule, only the ultrafiltration process takes place. Why? Because the glomerulus is a ball of capillaries which is located inside the Bowman's capsule, and blood is being filtered into this nephron, right? That means all our waste products, all the soluble products, right? For example, even glucose, even water, even our nutrients which are soluble, right, which are very tiny, they are easily filtered and they enter this nephron. Only the large particles like the red blood cells, white blood cells, large proteins like albumin, only these are being conserved in this glomerulus during the ultrafiltration process. Now we have a worry. If all our nutrients are getting into the nephron, that means they're about to get excreted. How are we going to prevent this from happening? We don't want our nutrients to be excreted. So we absorb them. We reabsorb them. This process reabsorb the imported water because a lot of water is being ultrafiltered. Therefore, we need to reabsorb this water. We need to take it back into our blood capillaries. So all these molecules, important molecules, are reabsorbed. But sometimes in this glomerulus, there could be toxic compounds like ammonia, right? Ammonia could be there. But sometimes our glomerulus might forget to actually put the ammonia into this nephron. Not forget, actually, sometimes the time might not be enough for all the ammonia to pass into the nephron. Because of that reason, some of the ammonia later they get secreted back into the nephron, and this process is called as secretion. Right, so that's a bit of a theory. Now you can see over here, they have labeled S and R. This S1 looks like a bit red for me, right? You don't have to know whether it's the artery or the vein, but you know they are the renal arteries and the renal veins, right? Renal arteries and the renal veins. And this is basically the not the urethra, the ureter, right? Ureter, which carries the urine to the kidney, to the bladder, right? Which carries the urine to the bladder like this. So here they're asking in the S1, what is this? So S1, which fluid is carried in the tube S? Which fluid? Because these renal arteries, renal veins, they're blood vessels. Blood vessels carries blood. So the answer will be blood, right? Answer will be blood, not bile. What is bile? Bile is actually a byproduct caused by the breakdown of red blood cells, right? It is produced in, it's formed by a pigment called bilirubin. It's formed in the gallbladder. It's formed in the liver and it's stored in the gallbladder. Later, it is being secreted into our small intestine, but that's not needed for now. And you can see filtrate. The filtrate means after the ultrafiltration, what is being filtered, you call it as the filtrate, right? So, the SNR doesn't carry the filtrate. And urine is carried by the Q, right? Which is our ureter. Therefore, the correct answer will be blood. Third one, they are saying, which tube is labeled Q in the diagram? It's the ureter. Urethra means the place where urine is passing finally out from your body. Ureter is the tube which carries the urine from these kidneys into the blood, right? Urethra. When you say urethra, this is how even when I was a student, I used to I get confused over ureter and urethra. When you say when you say through, it's like through, right? It's like through. T-R-O-U-G-H. It's like through. So you pass urine through your urethra, right? Something like that. You can like that's how I learned. That's how I kept it in my mind. That's like a tip to remember. Urethra you can learn that tra means through. So passing it through your body. You pass urine through your out. That means through your urethra to the outside, right? You can learn it like that. I don't know whether it would confuse you more, but it helped me a bit. That's why I'm sharing what I have learned with you, right? So you can use. So you can see that it is the ureter, because urethra is the one which you pass urine out from your body, right? Out from your body. Then B1, they have said analysis of the composition of urine can be used in the diagnosis of problems with kidney functions. Right, so they have given us three patients, and then they have taken the urine samples of these three patients, and then they have tested these urine samples for glucose content, for water content of the urine, and whether proteins are absent or present. So by looking at the results, by looking at the results, we have to determine what is the issue. Where is this issue? Where's the issue in the particular nephron, right? That means nephron is the tubule. Where exactly is the issue in that particular nephron? By looking at the results, we have to determine that. So you can see in the W patient, it says that glucose is absent, right? In the nephron, that means in the urine, glucose is absent, but the water content is normal and proteins are present. Do you know, if it's a normal urine, proteins should be absent in that particular urine? What is the reason? Why? Because if you take the Bowman's capsule like this, and if you take the glomerulus like this, here ultrafiltration take place. I already told you, during the ultrafiltration process, the proteins are not getting into this nephron. What is the reason? Because they are very big molecules. They can't diffuse into this nephron easily. Only the soluble ones, the tiny ones, pass through this membrane, right? It's a simple epithelium. They pass through this epithelium into the nephron. Therefore, large molecules like proteins, blood cells, platelets, they can't go inside the nephron. So if proteins are present in the urine, means there is some issue with the ultrafiltration process of that person. So the issue is ultrafiltration, right? So they're asking us to mention what is the process which is being affected. So you know the process which is being affected is ultrafiltration. And where exactly this ultrafiltration takes place? It's better to write it takes place from the glomerulus into the Bowman's capsule, rather than just saying it happens in the glomerulus, because you know it happens using both of these, right? Glomerulus as well as the Bowman's capsule. So it's always better to mention both of them, from the glomerulus into the Bowman's capsule. So that would be the exact location. Right. So the process is ultrafiltration. Location is from the glomerulus into the Bowman's capsule, and they're asking proteins are passed from the blood into the nephron during the ultrafiltration process. Yes, it's better to explain as well, because they're saying discuss, right? It's better to explain this rather than just writing the process and the location. It's better, even though they have asked the location and the process on it, it's better to explain this a bit, since they're using the word discuss. If you take your syllabus, right, in any Cambridge syllabus, sorry, not in any UK syllabus, you can see they give this word discuss, the word describe, right? So depending on those, the answers would be a bit different, right? I will discuss with that means I'll take that separate paper and I'll discuss it with you in our next day, or in the Christmas week. It's a bit safe. I'll be prepared by then, right? So on that day, I'll be discussing about the word discuss as well. After doing the paper, I'll quickly tell you what those things are, so you have an idea about those as well. So when they say discuss, it's always important to tell, that means to say something more rather than just the important points. So make sure that you write proteins are passed from the blood into the nephron during the ultrafiltration process. That's why if ultrafiltration did not take place, you can see proteins in the patient, in that W patient, right? Did you understand that?
Yes, miss. I also have a question.
Yes. Ask me.
So you said like when you're writing the location that the location where there's a defect as a glomerulus from the
Yeah, the
glomerulus into the
discuss.
Bowman's capsule. I wrote that
Yeah.
the process there's a defect in ultrafiltration because the blood that flows into the afferent arteriole contains proteins that must have bypassed the basement membrane of the Bowman's capsule.
Afferent arteriole. Yeah, afferent artery. That means you mentioned afferent arteriole and Bowman's capsule in your answer, right? You
Yeah.
didn't mention glomerulus. Yeah, because in the, I remember in the marking scheme, they gave marks even if you have written glomerulus only, or even if you have written Bowman's capsule. Either one, you get marks, right? And this was,
Oh,
this answer was also given. So if you have written Bowman's capsule, it's good. But don't, that means now it's okay. Next time when they ask about ultrafiltration, don't, that means don't write about the afferent arteriole, right? It's better to avoid the arterioles, efferent and afferent, and it's always better to talk about the glomerulus as a whole, right, rather than talking about the arterioles.
oh,
Okay.
yeah.
All right. Otherwise the answer is okay, but Bowman's capsule, you can get the mark, cut down that afferent arteriole part, right? Because
Yeah.
then that would make it more complex, and also it would, that means the right point is not there if you mention the afferent arteriole, because from afferent arteriole, ultrafiltration does not take place, right? That means, did you mention that it moves into the afferent arteriole after ultrafiltration or something like that?
Yeah, the blood that flows into the afferent arteriole.
Okay. No, then no issues, right? I heard it like I heard it like that from the afferent arteriole it passes into the Bowman's capsule. No, it's okay. You can write it like that, right? No
Yeah.
issues. Okay. Then you can say in the patient X, right? The glucose level, it's present, glucose is present, but water content is normal and proteins are absent. So glucose, anyway, you know, during the ultrafiltration process, glucose enters the nephron. But glucose doesn't remain in this nephron. Yeah, glucose doesn't remain in this nephron for a long time. Why? Because that's selectively reabsorbed back into our blood. But if glucose is present in our urine, that means the selective reabsorption process has not taken place, right? Therefore, you need that the process affected will be selective reabsorption. Due to that only, we will still have glucose in our urine. And where does selective reabsorption take place? It takes place in the convoluted tubules. You know, if you take the nephron, these two are the convoluted tubules. This
Yeah.
particular structure over here, yeah, the U-shaped structure, it is the loop of Henle, right? These two structures are the convoluted tubules. So you can mention the location is the convoluted tubules. And you can further explain it like this: glucose is not taken back into the blood from the tubule. That means this glucose, they're not taken, you know, surrounding this loop of Henle, there are some capillaries like this called as vasa recta. You don't have to know that there are some capillaries called vasa recta. So actually, glucose pass into these capillaries, into the blood in these capillaries. So actually, if the selective reabsorption does not take place, that means glucose is not taken back into the blood from these tubules, right? So if that is explained, you can get the, that means 1, 2, 3, 4. Yeah, if you have any 5 points, you can get the 5 marks for this question, right? No worries about that. But it's always, that means if you have these 1, 2, 3, 4, 5, the what you call the process and the location, and if you have written a description about that, you can get the 5 marks, right? Right. Let's move on to the patient Y. You can see in the patient Y, the glucose levels is absent, which means it's healthy. Proteins are also absent, which means it's healthy, but the water content is very high. Water content high means if you take the urine, the reason why the water content is high is because water reabsorption has not taken place. Water is not reabsorbed back into our blood capillaries. What is the reason behind this? You can see here they have said the process affected is reabsorption of urine or ADH release. ADH release means, did you know? Yeah, this ADH means antidiuretic hormone. When this ADH is there, right? When this ADH is there, what the collecting duct becomes more permeable. The collecting ducts become permeable and it will allow water to be reabsorbed, right? It allows water to be reabsorbed into the blood. But because the ADH release is affected, because the ADH is not released properly by the pituitary gland, due to that reason, the collecting duct is made impermeable to water, and water can't move out of this collecting duct. As a result, water will be there in our urine, right? So the full answer will be, you should mention that the process affected is ADH release. Better to write ADH release. And where does the ADH release gets affected to the collecting duct, because from the collecting duct only water is reabsorbed back into the blood capillaries. That is the main location, and that is the location where ADH also acts on. So what happens when enough ADH is not there? The duct is made impermeable to water, and it doesn't allow water into the blood, water moving into the blood. So if you have written this answer, if you have written 5 points, you can get the 5 marks. Right. Okay. Now, I have given a description of the role of ADH. Just to make sure that you have an idea about that. So you should know this ADH is basically secreted by the pituitary gland, right? It's actually, now for example, if you take too much of, that means, let's say you're eating that means you're eating fast food at night, right? You're eating a very heavy food with a lot of salt at night, and you're going to sleep. You might have experienced this. In the middle of the night, you suddenly wake up searching for water, right? That the reason is because, your body is carrying out osmoregulation, and ADH is released as a result. ADH can do 2 things, right? For you, it's better to learn this only. ADH actually, it makes our collecting duct more permeable. And the reason why you woke up was because hypothalamus directly sends signals to your brain to wake up, to drink more water, right? Because you have eaten, you have eaten some food with a lot of salt. In order to make sure that your body has enough water, only your hypothalamus sends signals to your brain to wake you up. That is a different process. Again, if you didn't wake up in the middle of the night, this is what hypothalamus do. Hypothalamus then will send signals to the pituitary gland, and the pituitary gland will secrete the hormone called antidiuretic hormone, or ADH. And this ADH hormone makes the collecting ducts permeable. So let's say we were going to excrete a lot of water from our urine, right? That's going to be a waste, right? We could have just used it up for different purposes, right? So we could, when ADH makes the collecting duct permeable, this water can be reabsorbed back into our blood capillaries. So water is retained in our body. That means we have enough water. So hypothalamus doesn't have to send signals to your brain to wake you up. It has already done its job by now. It has sent signals to the pituitary to release ADH. But what if ADH was not secreted? Then you can get, that means if ADH is usually secreted in a healthy person, but if it's not secreted in the correct amounts, or if it's not too fast for it to quickly act on the collecting duct and make it permeable, then your hypothalamus will send signals to your brain and ask you to wake up, right? So basically, here they have mentioned about the role of ADH. I'll send you a photo of this so you can read this as well, right, in your free time. Right. Let's move on to this part. Describe how to test for glucose in a sample of urine. This test for glucose is called as the Benedict's test, or else you can call it as, Benedict's test is the name. Here you use the failing solution, or simply you can call it the Benedict solution. Both names are the same, right? Failings as well as the Benedict solution. And to this solution, instead of writing the names, you can write down what is the composition of the solution. The composition is, there is copper sulfate as well as the sodium carbonate. Copper sulfate and sodium carbonate are there. So first of all, you take a test tube, right? And then to the sample of urine, you add the failing solution or the Benedict solution. Then, initially the color of the failing solution will be blue. Why? What is the reason? Because copper sulfate is there. Cu2 plus is there. Because of that reason, it appears as blue. But when you heat it, right? When you heat it, it will quickly change its color from blue to red. Why? Because it's been converted into Cu3 plus. Sorry, Cu plus is, Cu3 plus is not, Cu plus is being converted to Cu2 plus. Cu plus is blue, Cu2 plus is red, right? Wait, there is a bit of confusion over here. No. I'm sorry, Cu2 plus is being converted into Cu plus. Blue is being converted into red. Cu2 plus is blue, Cu plus is red in color. The blue solution is converted into red color. Why? Because, can you see, 2 plus is being reduced to plus. That's why we called glucose as a reducing sugar. Why? Because it reduces the oxidation number of copper 2 plus into copper plus. 2 is converted into 1. It is being reduced. That's why we call it as a reducing sugar. You might have heard the word before, reducing sugar, right? So glucose is a reducing sugar. As a result of that, the color of the Benedict solution changes from blue to red. But it's always better to write the series of the color change from blue to green, green to yellow, yellow to orange, because that's the observation that you see when you are doing the test, right? So better to write the observation, then you will get the full marks, right? So if you have any of the 3 points that I have mentioned, any 2 out of the 3 points that I have mentioned, you can get the 2 marks, right? Let's move on to the next question. Here they have asked us, here they have given us, here scientists have studied the population of two plants in two fields called X and Y. So they have given us a diagram as well, and they have asked us to calculate the mean number of plants of species B in field Y. So if you take species B in field Y, you can see 21, 26, 22 is there, right? When you count it at first, you get the count as 21. And then again the scientists come and then they again count it to make sure that they get a series of results to calculate the average in order to get an accurate result. Because of that, they again count it. The next time they get it as 26. And also in the third time also they count, and in the third time also they count and they get the result as 22. So you can see by looking at this, by analyzing this, you can see that count 2 is not very accurate as well, right? Count 2 is not very accurate. So yeah, count 2, that means 26. If you compare count number 1 and count number 3, it's close. 21 and 22 are close, right? But 26 is a bit deviated. I'm just saying for analysis, right? But anyway, since all these are close values, you can take all the 3 values, 21, 26, 22, and you can divide it by 3, right? You add them and you divide it by the number of counts you have taken. So at the end, you will get the mean number as 23 per meter square. So the answer would be 23 per meter square. Then they have asked us, describe how the scientist could determine the number of each plant species per meter squared in each field. So basically, this is something we all know. You can use quadrants, right? That's how they have counted them. By placing the quadrants on the particular grass or on the particular soil, they have counted the number of, right? They have counted the number of plants in each of these quadrants. So the first answer should be using quadrants, or else you can use random coordinates as well. And then you can count the number of species in each of these quadrants. You can use several quadrants and you can count the number of species in each quadrant. And you can divide the number by that area of the quadrant like this, right? Here they have divided the area. That means all this count means they have divided. That means they have divided the area. That means, for example, if this is 2 meters and if this is 2 meters, 4 meters per square, and let's say there are 32 plants, 32 plants per 4 meters square means there will be 8 plants per meter square, something like that, right? So basically you can determine the number of each plant species per meter square if you use quadrants. Last week also when we did the paper, if you have completed the paper, you come across quadrants in that paper as well. This quadrants is actually a famous part, right? Next time when we have time, I will be discussing a bit about the quadrants as well, showing you some pictures as well, because that needs a bit of a discussion, as these sort of questions are being repeated during the last 2-3 years, right? Now let's have a look at, here they are saying, discuss what the results show about the distribution of species in each field and the diversity between the 2 fields, right? So they're asking us to discuss. The word discuss is very important. They're asking us to discuss the results which shows the distribution about the fields and the difference in biodiversity between the 2 fields. So basically you have to discuss the differences between the 2 fields. As soon as you take a look at this table, can you see field X? It has 3 species. Look, all the 3 species has counts, right? But if you take field Y, this field Y, A species, if you take, it has 0 count. That means in field Y, the species A is not there, right? Field Y, the species A is not there. So the main difference is in field Y, in field X, you can see 3 species, but field Y has only 2 species. So that will be the main difference, right? Discuss what the results show about the distribution of species and the difference in biodiversity. So you can see X has 3 species and Y will have 2 species. That should be the first answer. Then the second answer will be, right? The second answer will be, you can say second answer, if you take the B species, if you consider the B species, this is how you compare. You have to go through all the values that they have given. You have to go through all the columns that they have given. If you take the B column, if you take the B row, you can see in field X, they have 5, 3, 13, and 7. They sort of have like an unequal distribution, right? Not sort of like an equal distribution. They have an unequal distribution. And also if you take 21, 26, 22, this has an equal distribution. This has an equal distribution. Again, let's go to C. 10, 13, 30. C in X has an equal distribution. And if you take Y, 1, 5, 6, sort of equal distribution. If you take A, 19, 22, 25, it also has a sort of equal distribution in it, right? So basically, you can write it like this. The field X shows little variation in numbers in species A, right? That means in species A, there is little variation, right? If you take the numbers, they're closer to each other. There's not much of a big difference because 19 and 22, 21, 22, 23, 24, 25, a difference of 2 or 3. Can you understand?
Yes, Miss.
All right. So the difference will be like 19, 22, 25, not much of a big difference, right? This is 19 to 22, you have around like 3. Again, 22 to 25, you have around like 3 values in between. So there is not much of a difference. So there is little variation in the counts that you have taken, right? So basically, you can say there is even distribution of species. Yeah. Yes. Yes. Or else, if now, since you have written field X, you can write it like this. Field X has 3 species. You get 1.4 right in that. But just because you write field Y has 2 species, you don't get another point. It's just the opposite of that. So better to write field X has 3 species. You can write it, but you won't get any mark. You won't get 2 marks for that. You just get 1 mark for this point. By looking at the field X, right, you can see that there is a larger biodiversity when compared to field Y. What do you mean by biodiversity? Diversity means the variation of species, right? Can you see in field X there are 3 different species, but in field Y there's only 2 different species. So in which field is the biodiversity high? In field X only, the biodiversity is high. So you can mention field X has a greater biodiversity, right? Field X has a greater biodiversity than field Y. Then you get another point in writing that. And then you can see, if you take the values, right? If you take the values, field X, species Y. Yes. Yeah, if you take the species B over here and over here, right? Species B, that means the number of species in field, the number of B species in field X is low, but the number of B species in field Y is high, right? This is sort of confusing because I understand that this is a bit confusing since they have asked us to discuss the results to show the distribution of the species. Did you understand these 2 points? Are these 2 points clear for you?
Yes, Miss.
All right. Can you let me know what your answer is? What the answer that you have written?
No, Miss, I wasn't getting it at first, so I just left it blank.
Okay. Right. So basically, these are the 2 points that you write, and the other 2 points, yes. How can I explain these other 2 points? These are just the counts, but they have given it as 12, uneven distribution. Shows little variation in the numbers. Okay, so basically, if you take this 23 and 24, you can see these values show little variation, right? The mean, that means you don't consider the counts, consider the mean, because that is the final number of species that you are considering. That is the most accurate value, right? If you consider if species A, it has 22 species. It has 22, not species, it has 22 individuals. Species B, it has 7 individuals. Species C, it has 12 individuals, right? That means field X, if you compare these values, they are a bit closer to each other. I'm not saying they are very close, but compared to 23 and 4 over here, they are close, right? That means 22 and 12 are a bit close. That means even 7 and 12 are a bit close, right? When compared to 23 and 4, there is a big difference between that, right? So you can say that there is uneven distribution in field Y. Why is that? Because B and C, the species B and C, they are not distributed evenly. Why? How do we say that a species is distributed evenly when there are almost equal number of species? When there are almost equal number of individuals in those species. But you can say in one species there's 23, but in one species it's just 4. So you can mention that there is uneven distribution, right? Uneven distribution in field Y, right? Uneven distribution in field Y. And over here you can see there is even distribution when you compare, there is even distribution in field X, right? Even distribution in field X, or else you can compare the values like this as well, right? You can compare the values like this. This is 12 and this is 4. That means the number of individuals in C species is 12 and the number of individuals in C species in field Y is 4. You can compare those values, and also you can say whether there is a higher variation or not. But it's better if you can just explain these ones rather than going into details, because that's what they have given, right? So I'll tell you what the final answer is. If you have any doubts, let me know. This is the final answer. 1 point for this, 1 point for this, and it's better to write that field X shows little variation in numbers. Little variation in numbers. Why is that? Because these numbers are closer to each other. I know they're not that close, but they're sort of closer to each other, right? But when you compare 23 and 24, so you can say field X shows little variation in numbers, and you can see there is even distribution of species within A and C, because A and C it's sort of even, right? Because 22 and 12 are closer to each other, right? That's what they are meaning over here. So you can write down there is even distribution between species A and C. Even distribution between A and C. But here you can even mention in field X that there is uneven distribution in B. Why? Because 22 and 12 are big numbers, but 7 is a very small number, right? Yeah. So you can mention about field X and write that down. Later, if you want, you can also write down in field Y, right? In field Y, there is that means there is a there is a big variation between the species B and C. If you have given those ideas, right, you can get the 4 points, right? Shall I repeat the 4 points?
Yes, miss.
Okay, so the first point will be field X has 3 species. Then the second one will be field X has a greater biodiversity. The third point will be field X shows little variation in numbers. Little variation in numbers. Field X shows little variation in numbers. Because actually we have to consider the results, not just the final value. Even if you take the results, field X, if you compare the values, can you see there's not much of a big variation? 10, 13, 13 if you take C, right? Even if you take A, 19, 20, 25. These are little variations, right? In any, you can consider it in the column manner or in the row manner, and you can take your answer, right? Even if you compare it in the row manner, you can see there is little variation, right? That means 10, 13 and 13, there's not much of a difference between these. 19, 20, 25, there's not much of a difference between the results that they have taken, right? Between the results. So basically, field X, the results of field X, we are not looking at the final field X. That means we're not looking at the real field X. We're looking at the result table. So the results in field X, it shows little variation, right? In numbers, right? Can you see? In A and C, it shows little variation. So write that down. That is the answer in the marking scheme as well. Field X shows little variation in numbers. Field X shows little variation in numbers. or in brackets you can write down even distribution of species A. Even distribution of species A. Then you can again write down field X shows little variation in numbers in species C as well. Field X shows little variation in numbers in species C as well. But if you take B, right, if you take B, it doesn't shows little variation. Can you see there is a high variation? 5, 3, suddenly a very big number is there. So there is a high variation in the species B in field X, because they are not asking us to compare. That means not, they are not always, that means even though they are asking the difference, they are just asking the difference in biodiversity. So we have already mentioned that field X has a greater biodiversity. We have answered that particular question. The discussing the result of the distribution of species is that means you have to discuss the results that you have got in this particular table. Did you understand that?
Yes, miss.
All right. If there is any confusing part, let me know that, right? So field X shows little variation in numbers in species A, and field X shows little variation in numbers in species C. In species C. Right. If you want, you can mention about these as well. You just have to discuss about the table, and at the end, you just have to say in which particular field is the biodiversity high. To support that answer, you have to mention that field X has 3 species, and that's why field X has a greater biodiversity, right? Okay. Let's move on to the next question. Name an abiotic factor that could affect the distribution of the plant species. Abiotic factors means the non-living factors, right? So non-living factors that can affect the growth of a plant, because it affects the plant growth only, the plants are distributed, right? That means because they affect, the abiotic factors affect the plants only, they are distributed in a particular manner. So the non-living things are temperature, water, sunlight, wind speed, mineral ions. If you have written any one of those, you can get that 1 mark. And also remember these for MCQs and all. It's better to remember these things. Right. Then you can see the diagram shows part of a molecule of DNA. Right. You can see it's a double helical structure. You call it a double helix. State what is meant by the genome. Genome means all the genes which are present in our particular, that means in our body, right? If you take an organism, all the genes which are present in our body is called as the genome. Or you can write down like all of the genes which are present in our body, or all of the DNA in an organism. Now there might be a confusion with what is DNA and what is a gene. So if you take DNA, it is a molecule like this. In the DNA, you have these what you call, these are called nitrogenous bases, right? Nitrogenous bases. These nitrogenous bases you can call, that means you might be calling it as a codon when it is being translated in, when it is being transcripted into an mRNA, but I won't refer to as a codon, because you call it a codon when it is in the mRNA. You don't call it a codon when it is in the DNA. So basically these 3 triplets, they are a gene, right? That means there are genes like this. They're triplets like this, right? Triplets. That means these triplets get together, and that is a particular gene. These gene codes for different things. Some genes code for our eye color. Some genes code for our hair color. Likewise, in a DNA molecule, we have all those genes, right? And there are some parts which do not code for anything, right? They are called as introns. They don't code for anything. They're called as introns. So those are not actually our genes. If you take the whole DNA molecule, not all the parts are genes. There are some non-coding areas which are called as introns, right? So in a genome, if they ask you what is a genome, that means all of the DNA in an organism is what you refer to as a genome. Or basically, it's better to write all of the genes in an organism. So you get 1 mark for that. Then they have asked to describe the differences between the structure of the DNA and the structure of the RNA. You know, when you say DNA is always a double-stranded molecule. But if you take RNA, sometimes RNA could be double-stranded, but because y'all are not learning that, y'all are only learning it as a single strand unit, it's better to say that RNA is a single strand. So, you know, DNA is a double helical structure with 2 strands, while RNA is just a single strand. That is the major difference. And you know, DNA stands for deoxyribose nucleic acid, right? And RNA stands for ribonucleic acid, right? Deoxyribonucleic acid means that DNA is made up of the deoxyribose sugar. Deoxyribose sugar. You know, a DNA is made up of a sugar like this, a pentose sugar, a nitrogenous base, and a phosphate group, right? That's how a DNA nucleotide is made up of, right? So DNA, what sugar does it contains? Deoxyribose. It's just the same ribose molecule, but it doesn't has an oxygen. That's why you say it as deoxyribose. But RNA, it contains ribose in it. And if you take DNA has adenine, guanine, cytosine, and thymine, right? You know, there are 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. You know that adenine forms 2 hydrogen bonds with thymine, and guanine forms 3 hydrogen bonds with cytosine, right? So have you, wait, give me a second. Yeah, so in your GCSE examination over here, can you see that they have mentioned it as G, C? Forget about this S and take this E into consideration. You can put this E in the middle and you can consider this as G and C are the 2 nucleotides, and this is the 3 hydrogen bonds. If you don't have a method to remember it, you can remember it in this way, but if you have already remembered it, it doesn't matter, right? It's quite easy, but for those are like simple techniques that you can use to keep those things in mind, right? Then you can see here, therefore you can see that the DNA will contain thymine, right? Instead of adenine, instead of uracil, it contains thymine. But RNA doesn't have thymine. RNA contains a nitrogenous base called uracil. And also, generally, if you take a DNA molecule, it is long, right? It's packed into chromosomes like this, right? It's packed into the chromosomes like this. But if you take the RNA, it's quite short. And that's why if you take the mRNA molecule, if you take the tRNA molecule, they're short molecules. They're not much long molecules. So therefore, DNA is longer while RNA is shorter. If you have written any of the 3 points that I have given over here, you can get the 3 marks. Then here they have asked us to describe the role of RNA in protein synthesis. The word describe is important because you can't just write down the points. You have to explain all these points in detail, right? I will quickly explain you what happens in, do we have time? Yes. Yes, we have time. Right, I'll quickly explain what happens in DNA in protein synthesis. So basically, if you take inside of a nucleus, you know that this is the nucleus, the cell is bigger than this. If you take inside of the nucleus, right? If you take inside of the nucleus, there is DNA in it, right? That means there is a DNA molecule. There are so many chromosomes, there are so many DNA molecules. So DNA molecule, if you take a double-stranded DNA molecule like this, during protein synthesis, this double-stranded molecule get converted into a single-stranded molecule like this, right? Then RNA nucleotides come and they create a mRNA molecule like this. They form hydrogen bonds with the DNA and they form a RNA molecule like mRNA molecule like this. This process is called as transcription, right? By looking at the DNA molecule, by looking at the DNA strand, they create a mRNA strand. This process is called transcription. And after this mRNA is created, you know there are nuclear pores. In the nucleus, there are pores. Through these nuclear pores, through these nuclear pores, the mRNA enters into the cytoplasm. The mRNA molecule enters into the cytoplasm. Afterwards, you know, in our cells, there are ribosomes, right? There are ribosomes. Actually, these ribosomes are made up of rRNA, right? But for this answer, you don't need that. There are ribosomes. What are ribosomes? The site of protein synthesis. This mRNA molecule comes and it gets attached with this ribosome. The mRNA molecule comes and gets attached with the ribosome, right? Then what happens? Yeah, then there is a pool called the amino acid pool in our cell. There are a lot of amino acids in this pool, right? So basically there is a type of RNA called tRNA, right? There is a type of molecule called tRNA. These tRNA molecules, they can combine with the amino acid because they have a structure called the anticodon. If you take the mRNA molecule, it has three nucleotides like this. Let's say AGU, adenine, guanine, and uracil, right? This AGU basically is one triplet. Why? There are three nucleotides. So this one triplet is called as a codon. Why is that? Because it codes for a particular amino acid, right? Usually AUG is the starting codon of any protein synthesis process. So AUG codes for the amino acid methionine, which you don't need to know, right? AUG codes for methionine. So basically, let's say this mRNA comes to this ribosome, and let's say this ribosome will read the mRNA. First, the ribosome will read the AUG codon. When the AUG codon is read, this tRNA molecule goes into this amino acid pool, get the methionine, it gets attached like this onto the head of the tRNA molecule. The methionine gets attached and it quickly comes into this ribosome area. So methionine will be there in the ribosome area. Likewise, when the ribosome is going on reading this mRNA molecule, several amino acids lines up like this and they create a polypeptide chain. So that's how proteins are synthesized. Later, these polypeptide chains, they get folded, they make different bonds, and they create a big compact molecule called as a protein molecule, right? So the answer should contain transcription produces mRNA because they're asking the role of RNA in protein synthesis. So it's better if you can write down what mRNA does, right? Transcription produces mRNA. How is this mRNA produced? Or what does this mRNA does? Transcription produces mRNA. mRNA. You have to mention the role of the mRNA. What is the role of the mRNA? mRNA, simple M, okay? mRNA copies the code of DNA strand. Copies the code of the DNA strand. And what does it do? It moves out of the nucleus. You have to describe. When you describe something, you say the whole process, right? So mRNA, so when you say that mRNA copies the code of the DNA strand and it moves out of the nucleus. You need to mention that. Moves out of the nucleus into where? Into the cytoplasm. Moves out of the nucleus into the cytoplasm, where it binds with the ribosome. Yes, where it binds with the ribosome. Right. With a ribosome. Right. Then what happens? You know tRNA. Now mRNA's function is over. tRNA will bring amino acid to the ribosome. Right? Will bring the amino acids to the ribosomes. Will bring the amino acids amino acids to the ribosomes. To the ribosomes. And you know this tRNA molecule I told you, it has an anticodon like this. That means if you take the mRNA molecule I told you, it has triplets like this and you call them the codon. So AUG is the starting, AUG is the starting code of the mRNA. The tRNA will then have the opposite of A, that means A will form a bond with thiamine, I told you, right? But you know, thiamine is not present in RNA. So there will be U over here. Yes. And uracil means like thiamine. So thiamine will form a bond with A. Guanine, I told you to remember it like GCE. So it will form a bond with C. So this is the anticodon of that tRNA molecule. And that particular amino acid, which is called as methionine, will join. AUG amino acid called methionine will join. Something like that will happen, right? Not something like that. That is what happens. So basically, the tRNA molecules bring the amino acid and they form peptide bonds like that, and they create a polypeptide chain. So the full answer, I'll read it out. Transcription produces mRNA. Transcription produces mRNA. mRNA copies the code of DNA strand. mRNA copies the code of the DNA strand and moves out of the nucleus into the cytoplasm. mRNA copies the code of the DNA strand and moves out of the nucleus into the cytoplasm. Moves out of the nucleus into the cytoplasm, where it binds with the ribosome. Where it binds with the ribosome. Then tRNA brings the amino acids to the ribosomes. The tRNA brings the amino acids to the ribosomes. Anticodon of tRNA will bind with the codon. Anticodon of tRNA will bind with the codon. Translation produces polypeptide. This process of converting the mRNA molecule into the polypeptide is called as translation, because you convert mRNA into a polypeptide chain. That's called as translation. Transcription was copying the information present in the DNA onto a mRNA strand. That is what you call as transcription. So protein synthesis has two steps: transcription, translation. So I'll read the full answer again. Transcription produces mRNA. mRNA copies the code of DNA strand and moves out of the nucleus into the cytoplasm, where it binds with the ribosome. tRNA brings the amino acids to the ribosomes. Anticodon of tRNA will bind with the codon. Translation produces polypeptides. Right. Did you miss anything? You can
No.
let me know. All right. Let's go to the fifth question. The diagram shows part of the female reproductive system with the structures labeled. Name the structures A, B, and C. So I have taken a clear diagram as well to explain the structures. You can see these thin tubes, they are called as the fallopian tube. That is the place, you know, in the ovary only you have the eggs, right? You call it as the ovarian follicles. Basically, you call them as the eggs. These eggs, basically the ovarian follicles, they get released into the fallopian tube once a month, which is called as ovulation, right? So fallopian tube is the one which helps the egg to be carried into this uterus, right? And you can see this uterus is basically, in simple terms, you call it the womb. It is basically the area where the baby is kept after the egg and the sperm are being fertilized, and the baby is retained, the fetus. You don't say it as, you don't call it the baby, you call it the fetus. The fetus is retained within the uterus. And then you can see the cervix. The cervix has a mucus plug usually during the pregnancy, because to prevent the entry of germs and other substances into the uterus. And you can see the vagina as well. right? So they have asked us to label the structures A, B, and C. You can call A as the oviduct or the fallopian tube, right? Oviduct or the fallopian tube, oviduct or the fallopian tube. And B is our ovary. And C, you can see they have not labeled over here. They have labeled to this small opening that is called as the cervix, right?
Yes.
Cervix. Yes. And D, you know, it's this particular chamber inside the female reproductive system that's called as the uterus. So basically, uterus, now you know the sperm and the egg get fertilized in this fallopian tube. This is the egg, this is the sperm. They get fertilized inside this fallopian tube. Afterwards, afterwards they form a zygote. And then later they form a structure called the morula. Then later they form another structure called the blastocyst. Or finally, the blastocyst, it needs to get into this endometrium wall. Not enter, basically it needs to combine with this endometrium wall. Because combining with the endometrium wall helps in creation of the placenta. The placenta is created for the transfer of nutrients and the exchange of waste, the transfer of waste materials to and from the to and from the fetus body. So because of that, the placenta is created. For that reason, this wall of cells needs to get implanted into this endometrium wall, and you call it as the implantation process. So uterus is the site of implantation. Afterwards only the blastocyst gets converted into the fetus. You can see the distinct structures of the fetus, right? You can see the eyes, the nose, and everything in the the scan that you do, the ultrasound scan, right? So basically that is the fetus. Before that, it's a blastocyst. It gets implanted into the placenta. Not to the placenta, to the endometrium wall, and this creates the placenta, right? Placenta is the structure which allows the exchange of materials between the mother and the fetus. So the umbilical arteries, umbilical veins, all these are present that means within the placenta, right? So basically this is the site of uterus, is the site of implantation of the embryo. Since y'all don't call it the blastocyst, it's better to refer to it as the embryo. Site of implantation, yes, of the embryo. Site of implantation of the embryo. Not just that, I told you, after implantation takes place, placenta, that means it creates the placenta. So placenta grows in this, uterus. Placenta grows in the uterus. So that is another function, another thing which happens inside the uterus. The placenta will grow. And also, do you know that this uterus, the endometrium wall, they secrete secretions. They release secretions into this, that means into the chamber. Why is that? Because in the early stages, they don't have a placenta. Now, before the embryo gets implanted into this endometrium wall, it doesn't have a placenta to get the nutrients. So where does it get the nutrients from? It gets the nutrients from this glandular secretions of the endometrium. In the endometrium is the wall of the uterus, the thick wall of the uterus. It has glands. Several glands are there. These glands, they secrete nutrients into the chamber so that the developing embryo can receive the nutrients. But after the placenta is created, it doesn't any longer need this, these glands. It can depend on the placenta for the exchange of materials and nutrients. So basically, you can refer to the placenta, or either you can refer to the endometrial glands, either one that you like. You can mention that the uterus provides nutrients and remove waste from the embryo. I'll say that, provide nutrition and remove waste from the embryo. Provide nutrition and remove waste from the embryo. Provide nutrition and remove wastes from the embryo. Provide nutrition and remove wastes from the embryo. Right. Then the C1, here they have asked, the graph shows the changes in levels of hormones released from the structure B. Right, what is structure B? B is our ovary. Ovary secretes only two hormones. One is the estrogen, the other is the progesterone. Right? Then what is LH and FSH? LH and FSH are created by our pituitary gland, right? By our pituitary gland. They are not secreted by the ovary, right? Estrogen and progesterones are the ones which are secreted by the ovary. So actually, the hormone which has two peaks like this, that hormone is estrogen. The reason is because now before the 14th day is the day of ovulation, right? Ovulation. Ovulation means the egg is being released into the fallopian tube on that particular day, right? So before yeah, do you have any questions?
No, miss.
Right. So before ovulation, that means ovulation happens when the egg is mature enough to be released into the fallopian tube. So from day 0 to day 14, the egg is getting developed. The egg is growing, right? The growth of the egg is taking place. When the growth of the egg is taking place, the egg releases a hormone. This egg, while it is growing, it releases a hormone. This hormone is called as the estrogen hormone. That is why you can see the estrogen increases before the ovulation, because the egg is being developed before ovulation is taking place. But right after ovulation has taken place, it quickly decreases, because now the egg has been released. It has already finished growing. Now it has been released. Now it's not growing anymore. But do you know, inside the ovary, after this egg is being removed, right, after the egg is being released, it forms this follicle. Actually, inside the follicle only you find the egg. After the egg is being released into the fallopian tube, this particular follicle becomes a glandular structure. This glandular structure is called as the corpus luteum. Even this corpus luteum can secrete estrogen. Not only that, it secretes progesterone as well. Corpus luteum secretes both estrogen and progesterone. That's why even after ovulation, you can see another peak in the estrogen. Right. And also in the N1, in the N diagram, you can see before ovulation, there was no peak at all in the N hormone, right? The reason is because progesterone, this hormone is progesterone. It's not secreted during the times when the egg is getting matured. It's not being secreted at all. After the corpus luteum is formed only, it's being secreted. In our syllabus, we have only two structures which secretes progesterone, which is the corpus luteum and our placenta. Right, placenta means basically the endometrium. That means the uterus. That means the placenta. Placenta. Basically the placenta. The what you call corpus luteum and the placenta only secretes progesterone. The egg doesn't secrete the progesterone. That is why before ovulation you don't see any progesterone levels. But after the corpus luteum is produced, after the corpus luteum is formed, you can see high levels of progesterone are there. So they are asking, what is the explain the importance of the changes in hormone M and hormone N during the menstrual cycle. So M is estrogen. You need to mention that. M is estrogen. And what does M do? Or else you can just write, even without writing M is estrogen, you can write M increases the functions of M. But it's better to give this introduction what M is. Then you can write down estrogen. What are the functions of the estrogen? Estrogen, no, they're not asking the functions. They're asking the importances of the changes. You can say before the day of ovulation. Can you see before the 14th day, there is a very high peak, right? So estrogen increases and peaks peaks before ovulation. Due to this increase in, there is another hormone called LH, called the luteinizing hormone. That hormone, as well as due to the increase in this estrogen on the ovulation take place. This egg is being released into the fallopian tube as a result of the LH surge and as a result of this estrogen surge. So estrogen increases and peaks before ovulation, right? Before ovulation. Right. So basically, that means it by this sentence they mean that it contributes to the ovulation, right? That's what you need to mention. Then you can write down N is progesterone. N is progesterone. For writing that, you get 1 mark. N is progesterone. Then you can write down what N does. N has a lot of functions. Did you know this progesterone, it maintains the uterine lining very thick. It maintains the uterine lining very thick. That means it prepares the uterus for a pregnancy. Every month it does this. The progesterone hormone prepares the uterus for a pregnancy, right? So what if the fertilization does not take place? What if the sperm and the egg did not meet, and what if fertilization did not take place? Then the corpus luteum that was formed, it get deteriorated. The corpus luteum which was formed get deteriorated if fertilization did not take place. As a result, will progesterone be secreted? No. Corpus luteum was the structure which was used to secrete progesterone. But if corpus luteum got deteriorated, no progesterone will be secreted. And no progesterone means this uterine lining will not be maintained. It will be broken down. This uterine lining is highly vascularized. Vascularized means it's filled with blood vessels. So basically the uterine lining breaking down means the uterine lining, the blood vessels are also being broken down and it is being released. So that's what you call as menstruation, right? So that's what happens if the corpus, if fertilization did not take place. So the importance of progesterone is it repairs the uterine lining, or it maintains the uterine lining, right? It maintains the uterine lining, or it repairs the uterine lining before, that means after menstruation also you need to build this again, right? After menstruation, again every month you have to repair this. You have to build a new one. For that also you need the progesterone. So you can write down N repairs the uterine lining. N repairs the uterine lining. Not just that, it also maintains it. N repairs the uterine lining. And also you can write down it inhibits, there is another function called as inhibiting FSH. It inhibits the hormone called FSH, follicle stimulating hormone. That's why during that is why during a pregnancy, right? During a pregnancy, another egg is not being matured because it inhibits the FSH, follicle stimulating hormone. FSH means follicle stimulating hormone means follicle is stimulated to grow. The growth of the follicle is inhibited, right? The growth of the follicle is inhibited in that case. When there is a pregnancy, because when there is a pregnancy, progesterone levels are high. Due to that, it inhibits the FSH levels, right? As a result, the growth of the follicle is inhibited. So you can write down it inhibits FSH as well. It inhibits FSH as well. That's another point, right? But anyways, if you have written those 2 points, it's enough for now, right? I will discuss these questions as well, right? It doesn't matter about the time. Let's quickly discuss this question as well. Other hormones are involved in the menstrual cycle. Describe the role of one other named hormone in the menstrual cycle. You know, luteinizing hormone is there. Also, follicle stimulating hormone is there. Sorry for the noise. I'm living in my boarding house at the moment. I'm extremely sorry for that noise. You can see the lutein. Yeah, okay, thank you. Luteinizing hormone is there and follicle stimulating hormone is there. These are the other hormones which regulate the menstrual cycle. So you have to mention one hormone, and you have to mention you have to describe the role of one of those hormones as well. If you take FSH, right? If you take the follicle stimulating hormone, what does it do? I already told you. It causes the growth of the follicle in the ovary, right? It causes the growth of the follicle in the ovary. Growth of the follicle in the ovary, right? Not just that, the FSH hormone also releases estrogen, right? FSH hormone also releases estrogen. FSH causes the growth of the follicle in the ovary, and it also releases estrogen, right? It's better to I think it's I think I made a little bit of a mistake over here. Actually, it's estrogen which repairs the uterine lining. M is the one which maintains the uterine lining. Okay, the estrogen repairs the uterine lining. M maintains the uterine lining. M maintains the uterine lining. Right. The estrogen is the one after menstruation takes place. N is the one which builds up the uterine lining, because you can see after menstruation, it again starts from day 0. Then you have only estrogen levels, right? So estrogen will build up the uterine lining, but after the 14th day, M will maintain that uterine lining, right? That was a small error which I have made. M maintains the uterine lining. Anyways, you can see FSH causes the growth of the follicle in the ovary. Not just that, it also releases estrogen. The follicle stimulating hormone releases estrogen. But did you know estrogen inhibits FSH? Estrogen will inhibit FSH, but FSH will stimulate the release of estrogen. That's an important point, right? That is why this estrogen is maintained at a low level over here at first, right? FSH, now basically FSH stimulate the release of estrogen. So at first, it's not there in the diagram, I'll tell you this. FSH is secreted in very high amounts at first. So we are supposed to have very high levels of estrogen, right? But over here in the diagram, over here in the diagram, we can see only very low levels of estrogen. The reason is, now even though FSH stimulate the release of estrogen, estrogen will inhibit FSH. Therefore, there will be little FSH. Due to that reason, again, there will be little estrogen. That is why the levels of estrogen are maintained low, right? But later, there will be a sudden peak due to the LH surge. Right? Due to the LH surge, there will be a peak in the estrogen as well, right? Did you understand that estrogen and FSH connection? That is important.
Yes,
right. Okay, so you can write down FSH. FSH stimulate the release of estrogen. FSH stimulate the release of estrogen. Those are two functions of FSH. And LH, two function is, LH mainly causes the ovulation. More than any other hormone, there is a very, now even though here they have given estrogen as a very big peak, if you compare the LH peak, that is going to be really high, because LH peak is the one which causes the ovulation to happen, right? LH causes ovulation. LH, due to the LH peak only, due to the LH surge only, there will be ovulation taking place. And also, after ovulation is taking place, the follicle should be converted into a corpus luteum, right? This is also this is also done by the LH hormone. Not only does it causes ovulation, but it also forms the corpus luteum, right? It also forms the corpus luteum. So if you have written any one of, that means if you have written one hormone and if you have mentioned one of the functions, you get 2 marks for writing that, right? Now let's quickly move on to the 6th question. Here they have given us a graph, and basically it's a bit of a complicated, it looks like a complicated graph, but the things that they have asked us is not that hard. You can see they have asked us to determine the difference in the percentage change. All right, percentage change means they have already given us the percentage Number of cases of measles, percentage change. Okay, they have only given us the number of cases of measles. We have to convert it into a percentage. That means percentage change in the number of cases between 1950 and 1965. Okay, it means over here and here we have to get the what do you call, the average. That means the number of cases, the number of measles cases over there, and we have to also find the number of cases between 1968 and 1983. That means over here we have to find that one also, that value as well. Finally, we have to calculate, we have to take the percentages, and finally we have to calculate the difference in the percentages. So first of all, let's find the percentage value over here. That means from 1950 to 1965. 1950, if you take the initial number of cases of measles, right, you can say it's 400, right? 400. If you see that, it's 400. But sometimes some people might see it as 420, right? So they have awarded marks even if you have taken 400, or even if you have taken 420, they have awarded marks. So in 1950, the number of cases will be 400. If you take 1965, the number of cases in 1965 will be around like 520. If you have got around like 510 to 520, you get marks, right? I'll take it as 520. So now we have to find the difference, right? We have to find the, that means we have to find the difference. We have to find the percent. That means the difference between 1950 to 1965. That will be the final value. Subtract the initial value, 520 subtract 400, and then you're finding a percentage. When you're finding a percentage difference, you divide it by the initial value, right? You divide it by the initial value. So 520 subtract 400, divide by 400, you multiply it by 100 to get the percentage difference over here. The percentage change over here between 1950 and 1965. That will be around like, you get the value around like 30%, right? But if you have got a value between 21% to 30%, you are correct, right? But by taking the values that I have taken, if you have got 30%, then that is also correct. Then let's see from 1968 to 1983, what is the percentage change? Because they're asking the difference in the percentage changes. So here also we are calculating the percentage change. Yeah, and here also we are calculating the percentage change. So let's find the difference in the percentage change from 1968 to 1983. 1968, it would be like 480 here, somewhere over here, right? 480 or 490. 480 or 490, I'll take it as 490. And 1983, it's like 120, somewhere over here, 120. So in final value is 120, right? The range is 120 to 130. The other range is 480 to 490. So if you have given any value between those values, then you are correct. Then how do you find the percentage difference? Final, subtract the initial. 120, subtract 490, divide by the initial, which is 490, multiplied by 100, you get minus 75.5%. You get a minus value. It doesn't matter. Then find the difference. How do you find the difference? Always the initial one, sorry, the final one, subtract the initial one, right? Since they're asking the asking the difference between 1950 and 1968, let's go with let's take the 1951 first. The 1950 to 1965 percentage change is 30%. Then you subtract. Then you again put the subtract sign and put 75.5%. Why did I take 30% first? Because they are asking the difference between 1950 and 1965. They have said this year range first. So I'm taking the answer of that year range first. Did you understand why I took the answer of that year range first? Rather than subtracting 75.5 from, that means rather than doing it like this, why I took 30% in front and I subtracted, right?
Yeah.
Okay, so 30% subtract minus 75.5%, you get 105.5% as the answer. That is the percentage difference, right? Between the number of cases Okay, so basically you have to discuss the relationship between the number of cases of measles, right? And also the percentage of 5-year-old vaccinated. Can you see after the, now from here only, the 5-year-old people, that means the 5-year-old baby starts to get vaccinated. Can you see there is a sudden drop from here to here? There is a sudden drop in the number of measles cases, because you know that children, they're subjected to a lot of diseases very quickly because they don't have a strong immunity, right? Because of that reason, if we can vaccinate them, right, because from children to children only it's quickly spread. So if we can vaccinate the 5-year-old children, then the chances of spreading that disease to other children is very less, because they are immune to that disease. They won't get that disease again. So if you vaccine the 5 rather than vaccine the elder ones, if you can vaccine the younger ones, because from the younger ones on it's been spread, right, very easily. So if you can vaccine the younger ones, then you can see the number of measles patients, the number of the cases of measles, they have suddenly dropped. So as soon as they have introduced the measles vaccine, and you can see the percentage of 5-year, or as soon as the percentage of the 5-year-old people are vaccinated, you can see a sudden drop. Can you see there is a big variation over here? But if you consider from here to here, from this level, even after the vaccine was introduced in this case, you still could see high peaks. But after the 5-year-old was started to get vaccinated, the levels drop very drastically, right? So that's what you need to mention. You have to mention that the cases are high. First of all, you can mention that the cases are high before the vaccine is introduced. You can mention this part also. The cases were high before the vaccine was introduced. So mention that.
Yeah.
Cases are high before the vaccine was introduced. Cases are high before the vaccine was introduced. Vaccine was introduced. The next point is cases are more variable as well. Can you see sudden at 1 year the cases are very high, the other year the cases are very low? That's because there are different varieties of that means measles. That means it could be caused by different varieties, right? Different variants, right? Because of that, that means it's caused by a virus, right? So there are different variants. Even COVID virus has different variants, right? So due to that reason, sometimes now after you get the virus, you get immune to that particular virus. That's why, that means after you get COVID-19, that means if you got COVID-19, that means if you have got COVID-19, I'm not talking about the vaccine, if you have got that disease, the next time when you face it, you're not immune to that, and you can easily combat that disease when compared to first time, right? Just likewise, you can, and that means you're immune to that disease. But what if another variant of COVID enters you? Then you're not immune to that disease, right? Then that new COVID variant, the new COVID-19 variant is new for you. Likewise, the measles variants can also change. You don't have to mention about the variants at all. I'm just explaining you what is happening over here. You can just mention that the cases are more variable, right? I just told you the reason why the cases are more variable. At 1 year, they can be very high before the vaccine is introduced. 1 year, they can be after the medications are done, they are very low. And again, if medications are not being carried out, they become very high. So you can write down cases are more variable before the vaccine was introduced. You just have to write what you see. Cases are more variable before the vaccine was introduced. Before the vaccine was introduced. Before the vaccine was introduced. Right. You can write it down like this, as no antibodies were there, because before the vaccine was introduced, there were no antibodies in those patients, right? That means antibodies were not produced. So you can write down cases are more variable before the vaccine was introduced, as there is no immunity or no antibodies. No immunity is better, as there is no antibodies. Right. Right. You can see when the percentage of the 5-year-old vaccinated increases, right? When the percentage of 5-year-old vaccinated increases, the number of cases with measles decreases. You can mention that as well, because they're asking us to compare that mainly, right? The number of cases measles and percentage of 5-year-old vaccine. When the percentage of 5-year-old vaccinated increases, the number of measles decreases. And then you can say there is less variation. There is less variation. Right? There is less variation. Right. Are you good with that part?
Yes, miss.
All right. Then the C1, some children are receiving treatments that reduce the activity of their immune system. Yes, due to some diseases, right? They might be receiving some treatments that reduces the activity of the immune system. And yeah, these are called as immunodeficiency diseases. That means immunodeficiency diseases means not actually those diseases. If you take AIDS, for example, it's an immunodeficiency disease. That means our body becomes less responsive to those bacteria, pathogens, and everything. Our immunity becomes very less responsive. And sometimes, this is just one example of a disease. Sometimes due to different medications. For example, if you are having a cancer, if someone is having a cancer or something, that patient is taking medications. Due to those medications, their immunity system is lowered, or the immunity system is weakened. Due to these reasons, they can get different other diseases as well, right? So basically, some children which are receiving treatments, it reduces the activity of the immune system. So if we are going to vaccine them, it's going to be quite dangerous. Why? Because in vaccines, sometimes they contain the weakened live pathogen. For example, if you take the COVID-19 vaccine, it contains a dead COVID-19 virus, or else it contains a weakened COVID-19 virus. Why is that? Because when it enters, right, when the COVID-19 virus enters in our body, it can create the antibodies. Our body will create the antibodies to kill that COVID-19 virus. Right? Our bodies will create the antibodies to kill the COVID-19 virus. And also it's good. Why? Because this COVID-19 virus is very weakened. It can't do any harm for us. That's why after vaccination, in the first day, you can get fever and all, right? You get some side effects, but afterwards you're healthy. Right? So this COVID-19 is weakened. It can't cause any diseases to you. And also antibodies are created, and another type of cell called memory cells are created. That is the importance of vaccination. After you vaccine, that means after you get chickenpox, right? You won't get chickenpox again in your life, because the memory cells are created in your body. That means when another chickenpox virus enters your body, as soon as it enters your body, the old memory cells are there. They remember, okay, I remember you. You came previously as well. And they started killing this chickenpox virus very quickly. Right? So likewise, even in any sort of vaccination, it creates memory cells. But right, if you vaccine it to the children which has a very low immunity, which has a weakened immunity, the weakened COVID-19 pathogen, even though it didn't do much harm for us, it can do harm for these children with the weakened immune systems. Why? Because they have a weakened immunity. They can't create much antibodies to kill this weakened COVID-19 pathogen. right? So this
Yeah.
weakened COVID-19 pathogen can do a lot of effects, can have a lot of side effects on their body. That's why you should not use the vaccination on children having such kinds of medication. That's why you also have a certain age to get vaccinated, because sometimes small children, their immune system is not yet developed properly, so you shouldn't vaccine them. So the final answer for this question is, explain why vaccinations are not used in children. They cannot produce antibodies. If they are vaccinated. These are the points. I'm just saying the points. They cannot produce antibodies. That is one point. And also, few memory cells are produced. That is another point. Few memory cells are produced. That is another point. And they could develop that disease. Now, if we administer the weakened COVID-19 to them, they could get COVID-19, because that's, even though it's weakened for them, even though it's weakened for us, it's very strong for them, so they could develop the disease. That's the important point. So if you have written any of the 3 points, any two of the 3 points that I have given, you can get the marks. Then the final question, this is on micropropagation. I have given a note on micropropagation as well, right? So let's quickly read. Micropropagation uses small fragments of plants which are regrown into whole plants. These fragments of plants are known as what? Can you tell me what they are known as? Do you know the
Yeah,
answer?
explants.
Yes, very good. Explants. So if you take now, what are these explants? If you take the parent plant, you can scrape the cells from this parent plant, and you can take those cells and plant them in petri dishes in the laboratory, and you can allow them to grow. These are called as explants. The fragments of plants are called as explants. And then they have said the surface of each fragment is sterilized to prevent the growth of, can I know the answer?
Microorganisms.
Yes, it could be microorganisms. So you can be specific and you can write fungi, bacteria, pathogens, right? They have given marks for any of those. And microorganisms, it's good. So the fragments are placed in a growth medium. The growth medium contains agar and a source of energy such as
Starch.
Yes, starch, or you can just write sugar, or you can write glucose, or you can write sucrose. Starch is also good, right? This method is able to produce large quantities of genetically identical plants called?
Clones.
Identical plants called what?
Clones, miss.
Yeah, clones. Very good. Clones. Yes, clones, right? That means producing identical plants. It's called clones. One advantage is that micropropagation is quicker than?
Sexual reproduction.
Yes, sexual reproduction. Why? Because micropropagation is a type of asexual reproduction. There is no male, there is no female, right? There is only just one parent plant getting involved. So that sort of reproduction is called as asexual reproduction, and micropropagation comes under that. So it involves flowers and seed production. Another advantage of micropropagation is that plants can be produced
All the year or any time of the year.
Yeah, all the year or any time of the year, right? Since we have 2 minutes, I'll quickly go through this. Micropropagation, you can see it's the micropropagation. So basically it's tissue culture. It's a process where small pieces of plants are grown in a nutrient media, basically in a petri dish. You have agar, right? You have all the nutrients which is required for the growth of the plant, right? So all these things are there, and you have a parent plant. You scrape the cells from the parent plant. These cells are called as the explants. They are planted over here. See, these cells are called as explants. And you can see before they are planted, they need to be sterilized using a disinfectant, right? Why? Otherwise, that means even this, what you call, during this plant, that means when you're about to, that means your hand could contain germs, the air could contain germs, right? So the cells are newly opened cells, right? So they can easily get contaminated with the pathogens in the air and everything, right? So you should definitely sterilize them before allowing them to grow in the petri dish. And you can see the petri dish, the agar, the nutrient agar, all should be sterilized as well. And you can see they divide and they grow into small, they divide into small masses of cells known as a callus, right? And all these and you can see each callus is transferred to a fresh growth medium that contains a range of plant growth, so basically plant growth hormones. So basically after the callus is taken out, you can again put them into different petri dishes, and then you can add the relevant plant hormones. So the callus can differentiate into roots, stems, and leaves, and it can form a small plant. Later, you can again transfer that plant into soil if you need it, but usually they are grown in laboratories, right? In the that means in the it's called the horticulture. In the horticulture area, it's grown, right? So that is it for today. I hope that you have learned something from today, right? I'll be winding up for now. If you have any questions, you can ask me through WhatsApp about this paper. I will definitely answer. Even if you have any other questions on any other biology book, don't hesitate to ask me. I will answer those questions. And do let me know about the example of that plant that you told me about, the plant which has
I said
less.
it was water lilies.
Oh, water lilies. Oh, yeah, water lilies. Yes, I didn't know that. I have to check that. Is it in the Edexcel IGCSE book?
Yes, miss. There was a past paper question about it. I remember.
Okay, okay. I have to check it then. Right, right. Okay, so basically, I'll have to check it. The water lilies. That's good that you told me that. I didn't come across that. Right. So I hope that you have learned the what you call the new theories from today's lesson. Do let me know if you have any doubts. Yeah, I'll be winding up for today. Right. Thank you for joining. I'll be winding it up for today.
Okay, miss. Thank you.
You're welcome.